Hello guys, how are you? I hope you are well. Today's lesson is going to cover microscopy, which is 4.1.1.5 in the AQA specification. The idea behind today's video is we're going to cover the basics that you need to know for your GCSE science exam when it comes to microscopy. The four areas we're going to cover today is understand how microscopy techniques have developed over time, explain how electron microscopes have increased the understanding of subcellular structures, and that word subcellular is super important because ultimately it relates to things inside the cell, things smaller than the cell. So your nucleus, chloroplast, mitochondria, all of those things were visible for the first time using electron scanning microscopes. Express answers in standard form, and I'll talk about that when we get there, but this is something you've learned in maths and hopefully we can build on and develop and apply in science. And then finally carry out calculations involving magnification using the formula. As always, make sure you've got a pen, you've got a paper, something to write on, something to write with, and let's go. So the first microscopes, the early microscopes, were developed in 1590, and it wasn't until about 350 years later when in 1942 when the first electron scanning microscopes were developed. Now the idea behind this, or the differences in image, were incredible. If you look at the image on the left, that is taken from a modern day light microscope, which you probably have in school, which you probably practiced using for the required practical. And then on the right hand side I've got the image of a electron microscope, and usually those images are black and white, but they can be processed and colour can be added to those images to make them pop and to make them look really, really good. However, the key differences that you need to know are electron microscopes have a much higher magnification ability and they have a much higher resolution. Now those two things hopefully should be relatively straightforward. If you've got a higher magnification ability, it means that very simply, oh, pen's playing up, it means very simply that you can magnify in much greater detail the image that you're trying to look at, or the object that you're trying to look at. The other thing that you need to understand is electron microscopes have a much higher resolution. Now, resolution is the ability to distinguish between two points that are close together. If you've ever played really old video games, which is my go-to reference, if you've played any old video games, hopefully what you can see is that the resolution is very low. It's very difficult to tell details. Compare that to the modern day, and actually modern day games have much higher resolution and therefore they can add a lot more detail to the image. Now, ultimately, boys and girls, what you need to understand is, because of those two major differences, we are able to observe cells in much finer detail. And because we're able to observe, sorry, it's going to bug me, my OCD's getting to me, magnification, let's highlight all of the word. If you can see things in much finer detail, then you can observe things like subcellular structures, which I've kind of touched on already, so I'm not going to go through again. Now, this is a diagram of a pretty standard basic microscope, a light microscope. Again, like I said, which is what you've probably used in school, and I'm just going to talk you through three main parts in my opinion. The eyepiece lens, which is where you put your eye, believe it or not, so that's hence eyepiece lens is an easy name to remember. The objective lens, and we are talking about the object, and hopefully that should be straightforward as well. And then finally we've got the stage where you put your sample. Now, two things to draw your attention to. The eyepiece lens usually comes in a ×5 or a ×10 magnification, and the objective lens can come in a whole host range of different magnifications, anywhere from ×20 to ×400, even more, depending on the microscope that you have. Now, the key thing is when you set up your microscope, and you've hopefully done this in class, when you set up your microscope you need to have it in the lowest zoom combination, in the lowest magnification possible. The reason being is that it gives you the widest field of view. So when we talk about field of view, what we mean is what you are able to see. Now, the widest field of view, the best example I give my students is if you're playing a game and you're the sniper and you look through the scope, if you're zoomed in as much as you can be, then you're not going to be able to spot whatever target that it is that you're looking for. However, when you first put your sniper rifle scope on, you need to be zoomed out as far as possible in order to get the widest field of view. Then you pinpoint, right, that's where that person is. I need to zoom in that particular location, and microscopes work in a similar way. You zoom out as far as possible to decide this is what I'm trying to zoom into, and then you change your objective lens to get a bigger magnification. The total magnification of the microscope that you're using can be calculated using, or rather not using, total magnification can be calculated by doing the eyepiece lens multiplied by the objective lens. So whatever the magnification is for the eyepiece, multiply it by the objective. So for example, if I had an eyepiece lens that was times 10 magnification and I had an objective lens which was times 40, I would multiply the two numbers together to get a magnification of 400. Hopefully, should be really straightforward. If, I'll give you another example real quick, times 10 for my eyepiece, and my objective lens has a magnification of times, I don't know, 30. My total magnification is going to be times 300, and that's the magnification that I would look through my microscope. Again, bearing in mind, eyepiece lenses can only have a magnification of times 5 and times 10. So for the next part of the lesson, we're going to look at standard form. Standard form is a technique used in maths to simplify, or rather to write really large numbers or really small numbers in a simplified manner. So I'm going to give you two examples, and we're going to start with the ones at the top. So 2,340,000. Let's write that number down. 2,034,123 zeros. Brilliant. When I write standard form, the two numbers that I'm interested in really is the first number and any non-zero numbers that come after it. So 2.34 is what I'd write. However, I need to multiply it by a power of 10. In order to do that, I need to count the number of digits that I have after the first one. So if I look at this graph, if I look at this number here, I've got 3, 4, 1, 2, 3, 4 zeros. Overall, I've got 6 numbers after my 2. So I'm going to do 2.34 multiplied by 10 to the power of 6. That's straightforward. So 2.34 times 10 to the power of 6. And that is 2,340,000 written in standard form. Now, why do I multiply it by 10 to the power of 6? Well, if I do 234 multiplied by 10, I get 23.4. If I do 2.34 times 100, so 10 times 10, I get 234. If I times it by 10 again, I get 2,340. So actually, if I keep going and times it by 10 6 times, I would end up with 2,340,000, which is the number we had at the beginning. Oh my god, we got there. Let's clear the screen. Now, the second number is similar, but it's slightly different in the sense of it's got a lot of zeros before the first non-zero digit. So again, I do the exact same thing in the sense that I write down the first non-zero digit, which is 8.21. From this, what I need to do is say, right, how many zeros are there before the 8? So I've got 1, 2, 3, 4. So I do times 10 to the power of 4. But I have to be really careful. The reason I have to be really careful is because if I just did 8.21 times 10 to the power of 4, that's going to be 82,100. That's not what I'm after. That's not the number that's written down on the screen in front of you. Actually, I need to do 8.21 times 10 to the power of minus 4, and that means that it goes down in number place value. Maths teachers, I know you're ripping your hair out. Please forgive me, I know, but it's just the easiest way for me to explain it to my students usually, and therefore that's what I'm going with, and that's what I'm going to stick with. 8.21 times 10 to the power of minus 4. Again, 0.123 was the number, 8.21 was my non-zero digits, and then I've got 1, 2, 3, 4 zeros before it, hence where that comes from. Now, your turn. I want you to have a practice. I want you to pause the video and I want you to see if you can work out what the numbers should be in the table in standard form. I'm going to clear the screen to make it easier for you to process. No, I haven't paused, don't worry. I actually want you to pause though, so please pause, and on your marks, get set, go.